Welcome dear students. In this video we are going to learn about the last pathway of GPCR signaling. In this case the G protein coupled receptor activate the PLC beta effector and then IP3 and ultimately it produced nitric oxide. How it occurs I will show you. Let's suppose this is a plasma membrane. Specifically I want to mention here that this pathway actually occurs in endothelial cells. Endothelial cells that lines the blood vessels. It could be arterioles or veins, but these are the endothelial cells that actually line the blood vessels. So in this case, in this plasma membrane there is a G protein coupled receptor is there, and this GPCR bind with a molecule called acetylcholine. So acetylcholine when binds with this G protein coupled receptor that is present in the endothelial cells, it activates a special G protein that is called G alpha O. You can see here I have written here G alpha O. This G alpha O gets activated because it associates with GTP and helps, and this transformation is actually driven by this GPCR because it acts as a GEF. When G protein coupled receptor binds with acetylcholine, it actually functions as a GEF and activates this G alpha protein. And when this G alpha protein gets activated, it activates another effector protein that is called PLC beta. This is an enzyme. Then PLC beta convert PIP2, that means phosphorinositol diphosphate, into two products that is called IP3 and DAG. In my previous video I have mentioned that both this IP3 and DAG actually helps in this pathway to activate PKC, because IP3 increase the calcium concentration and DAG directly bind with PKC, but proper activation of PKC requires calcium also. I have talk about in my previous class. But in this case, in case of endothelial cells, the DAG is not very important, but the IP3 is very important. IP3 increase the calcium concentration in the cytoplasm. How? Because IP3 is actually a molecule that can bind with the IP3 gated calcium channels that is present in the endoplasmic reticulum. In the endoplasmic reticulum there is a channel which can bind with the IP3, and when IP3 binds, this channel actually gets open and the calcium that is stored inside the endoplasmic reticulum actually come out to the cytoplasm. When calcium concentration increases, this calcium actually bind with a molecule called calmodulin. So calcium binds with calmodulin. Then these two complex, actually calmodulin protein, activates another protein that is called nitric oxide synthase. This is an enzyme, nitric oxide synthase. It can convert a molecules called arginine associated with oxygen. It converts arginine and oxygen to citrulline and nitric oxide. Citrulline and NO, nitric oxide. This is the main thing. This is the second messenger here. This nitric oxide then diffuse from the endothelial cells to the nearby smooth muscle cells. Actually you have to understand this first. This is the blood vessels, okay? Let's suppose from here blood flows. This blood vessels is covered by, or actually lined by, endothelial cells, and this reaction actually occurring in these endothelial cells. Now these blood vessels, these endothelial cells and the blood vessels actually attached with smooth muscle cells. So this nitric oxide then diffuse from the endothelial cells to the smooth muscle cells. It diffuse to the smooth muscle cell. In the smooth muscle cells there is a receptor for this nitric oxide that is actually a soluble means it is present, this receptor is present in the cytoplasm, not in the membrane. This receptor is nothing but a cyclic GMP converting enzyme that is called guanylyl cyclase. So this receptor that actually binds with, let's suppose this green is the nitric oxide. When nitric oxide binds with this receptor is actually nothing but a guanylyl cyclase. Guanylyl cyclase. In this case, I want to mention that this guanylyl cyclase receptor can be in soluble form or in transmembrane form. In 2020 June CSIR NET examination, this type of questions has come. I will show you in my another video. So guanylyl cyclase is actually enzyme, acts as a receptor for nitric oxide that is soluble in conditions in the cytoplasm. Then this nitric oxide bind and activates this guanylyl cyclase activity. Then this guanylyl cyclase actually convert the GTP molecule into CGMP, cyclic guanosine monophosphate. Now the CGMP activates a protein called protein kinase G, PKG. Okay, the ultimate product is the PKG. This protein kinase G actually dephosphorylates the light chain of myosin so that it cannot contract and actually activates different proteins, and ultimate result is a smooth muscles relaxation. Smooth muscle relaxation. When smooth muscles gets relaxed, the blood vessels actually dilates. It is called vasodilations. So the ultimate result of this signaling pathway from the acetylcholine, and the last thing is the blood vessels vasodilations, called vasodilations. You have to understand this. When blood vessels get dilated, that means vasodilation occur, more blood actually flow through the blood vessels, and it actually helps in different functions. Like, I want to give an example. In case of erectile dysfunctions, where penis do not get erected, this process actually has any problems in those victims or patients. And another thing I want to mention here, how this disease actually treated. Let's suppose here that this CGMP is very important. I have mentioned here CGMP. You can see here the whole process, acetylcholine, then this G alpha protein activated, then this G alpha protein activates PLC beta, it cleaves the PIP2 to IP3 increase calcium binds the calmodulin, it activates an enzyme called nitric oxide synthase. Nitric oxide synthase produce nitric oxides then diffuse to the smooth muscle cells and activate a receptor that is actually nothing but guanylyl cyclase. Guanylyl cyclase converts the CGMP, and CGMP activate PKG. But there is a enemy of this CGMP that is called phosphodiesterase. Specifically phosphodiesterase 5. Phosphodiesterase 5, that is the target. Let's suppose only you remember phosphodiesterase. This phosphodiesterase actually directly destroy the CGMP to make GMP. So this is the enemy. In case of erectile dysfunctions, a molecule, actually a drug that target and inhibit these PD molecules, that is called sildenafil, or a branded name medicine called Viagra. So Viagra acts in this particular molecule of this signaling pathway. PD, it blocks the PD, so the PD cannot degrade the CGMP. As the CGMP remains high, it activates the PKG and it actually helps in the vasodilations. More blood can flow to the penis and it will erect. So this is the whole signaling pathway of the GPCRs. I have completed the 5th pathway also. In 2020 June examinations, one question has come that is related with this guanylyl cyclase, because I have mentioned that this guanylyl cyclase can be two form. One is in the membrane bound conditions. Let's suppose this is the plasma membrane. So guanylyl cyclase can be in a plasma membrane in conditions. This can be a CGM guanylyl cyclase, GC, but in transmembrane conditions. Another is in the cytoplasmic region. This is also function as guanylyl cyclase. In case of nitric oxide, it is in the soluble conditions. But in case of a protein or peptide called anti-natriuretic factor, atrial natriuretic factor, this factor or a protein, anti-natriuretic protein, binds with the transmembrane or integral membrane receptor, that also actually a GC, guanine cyclase. So guanine cyclase can be cytosolic or soluble, cytosolic or in transmembrane conditions. And there is a question that in this recent CSIR NET examinations has come. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching and always keep learning.